Hi, my name is Sarah Lehman, and I work for the United States Environmental Protection Agency. I'll be presenting information on a National Water Quality Monitoring and Assessment program called the National Aquatic Resource Surveys, or NARS. This program is a collaborative effort between EPA, states, tribes, and others to assess the condition of our nation's waters. During this workshop, I'm also excited to learn more about the work that you all are doing on recreational water issues and to further my understanding of how NARS data can feed into your effort and be leveraged to enhance the ability to address these critical human health issues. As I get started, I also want to recognize all of the field crews, the lab personnel, data managers, analysts, and other staff who dedicate so much time and energy to NARS. So today I'll be providing some background on the NARS, why we're implementing a national assessment, as well as highlighting a few findings from two of our recreational related indicators. I'll also present a few examples of how scientists have used the NARS data to address other questions beyond the basic NARS objectives to look at issues related to recreational concerns. And finally, I'll provide you with some links if you'd like to explore our data or results in more detail. So why a national monitoring program at all? We know that the U.S. spends significant resources to address water quality. Congress recognized the need for monitoring to support Clean Water Act efforts by requiring states to have monitoring programs, as well as including implementation of a national surveillance system in the Act itself. However, when leaders, such as former US EPA Administrator Riley, were asked the seemingly simple question of whether water quality was getting better or not, they've had to acknowledge, as he did in 1989, that they didn't have the answer to the question. A number of critiques were also published during the 1990s and early 2000s that further highlighted inadequacies in our collective monitoring programs to provide data in support of the various needs of water managers from local to national scales. In response, EPA worked to implement a monitoring initiative supporting enhancements to state and tribal monitoring programs and national assessments. Today's NARS program is the result of a significant amount of scientific research by EPA's Office of Research and Development in collaboration with states that established the technical foundation for the program. And it's also a result of EPA, OMB, and Congress recognizing the applicability of statistical survey approaches as a cost-effective means of addressing a portion of the identified monitoring gap. NARS is specifically designed to assess not individual waters in detail, but to provide information across the population of waters for our coasts, our lakes, rivers and streams, and wetland resources. As a program, we've had three overarching objectives. First, to assess the biological and recreational condition of the nation's waters and to track changes over time. As we're starting to progress into the third and fourth iterations of our surveys, we're also increasingly able to look at trends. Second, the program is designed to rank stressors in terms of their impact on condition. And third, the program aims to enhance state and tribal monitoring and assessment capacity. To date, we've undertaken 11 surveys, starting with lakes in 2007, through our most recent adventure, which was implementing the coastal survey last summer during the pandemic. As a result of the pandemic, this summer we'll be working with our partners to finish the coastal survey, as well as implement the third wetland survey. A significant component of the NARS is achieving and maintaining national consistency in the assessments for each of the water body types. To address this, NARS employs a statistical survey or probabilistic design, where sites are selected randomly across the country, allowing us to extrapolate the results beyond the site sampled to the entire population of waters. For example, for the recently released Rivers and Streams report, our partners sampled nearly 2,000 sites over 2 summers across the conterminous United States, but the results themselves represent about 1.2 million miles of rivers and streams. So our current implementation strategy has us rotate between the water body types each year. So last summer we were focused on coastal. This summer we're implementing the wetland survey along with finishing up coastal. In 2022 we'll be sampling in lakes, and then we'll return to rivers and streams. For NARS, across the country, all the field crews use the same methods to collect the data, and the same data are collected at all sites for each water body type. Similarly, standardized lab protocols are used. We also employ a national quality assurance and data management program to support consistency in those data sets. And finally, we develop national and regional scale assessments and produce peer-reviewed reports summarizing the information. During this presentation, we'll be focusing on results for 2 indicators, microcystins and enterococci, but also wanted to note that for each survey, we include a number of other important chemical, physical, and biological indicators. All of the indicators for each survey are co-located, allowing for researchers to conduct a variety of analyses looking at relationships between condition and stressors, as well as the variable lake landscape. Some of the indicators we include are noted here on the slide. For example, we collect data on benthic macroinvertebrates and phytoplankton in lakes, vegetation and wetlands, and nutrients in all 4 surveys. We also include various research indicators such as microplastics or environmental DNA work that may or may not become part of the core indicators that are repeated over time. So as I move into showing results from the national surveys, I wanted to mention some points on how we collect algal toxin and enterococci samples. Our design is not aimed at tracking harmful algal blooms or collecting samples specific to beaches or other recreational areas. We collect these data at all of the randomly selected sites in the country, and they're collected as part of our typical implementation process. Samples are collected during our summer index period. We typically collect just 1 sample per site, which allows us to look at occurrences spatially to see whether there's occurrences in time. Samples are collected near the surface of the water, and crews are also asked to avoid scums when possible. So taking that into consideration, I'll provide some national and eco-regional results for these 2 indicators. For microcystin, we used ELISA kits to analyze samples to provide screening level assessments. For the most recent surveys, we've rarely seen exceedances of the human health benchmark established by EPA, 8 micrograms per liter. There are some differences in the percent of waters where microcystin are detected, ranging from more than 30% of lakes, river and stream miles, and Great Lakes shoreline area, to 12% of wetland area, and just 6% of estuarine area. Although this is a naturally occurring substance, I still think it will be interesting to track whether detections or exceedances change over time and whether any changes seen can be linked with changes in stressors such as nutrients or climate change variables. Also, while not shown here, we've collaborated with USGS to assess a broader suite of algal toxins as part of the Coastal 2015 survey, and we started including cylindrospermopsin for some of the surveys. For enterococci, water samples were analyzed using qPCR. Results were compared to the EPA recommended water quality criterion for protecting human health, the 1280 calibrator cell equivalents per 100 mils. In this case, we found a greater percent of river and stream miles with exceedances at about 30% of miles than we did with the other water body types. Lakes came in second with about 4% of lakes. The NARS data can also be presented at different regional scales. So in this case, I'm showing aggregated ecoregions. And focusing in on the river and streams enterococci information some more, we can see that there are also differences in terms of exceedances across the country. Generally speaking, the percent of river or stream miles exceeding the benchmarks were lower in the western part of the country than in the central and eastern part of the country. And if you can focus in on the graphic, you'll see that they range from about 13% of rivers and streams in the xeric and northern plains ecoregions, so the yellow and purple on the map, to about 39% in the temperate plains and coastal plains, so more of the central part of the country. And then turning from the national and ecoregional presentation of primary NARS results, I also wanted to touch on a few examples of how NARS data have been used to address other research questions or programmatic needs. First, and most recently, data from the lake survey were used in the development of EPA's draft lake nutrient criteria for aquatic life and recreational uses. NLA phytoplankton, zooplankton, nutrient, chlorophyll A, and microcystin data were all used in development of the models. Other examples include work done by Beaver et al. In 2 papers that looked at environmental factors associated with potential toxin-producing cyanobacteria in lakes across the country. They found that land use factors are related to cyanobacteria and that microcystins are associated with areas of low forest cover and high nutrients. Another example is Leach et al., in which the authors identified an increase in murky lakes across the country and surmised that could be leading to lakes that are more susceptible to nuisance algal blooms. 2 other examples include papers looking at continental increases in phosphorus levels and work to predict streams that are good candidates for protection and restoration efforts. And then finally, I just wanted to provide some information on where you can go to access tools from the program. 3 of our surveys currently have dashboards available where you can explore assessment results in more detail. This includes looking at the data by different subpopulations such as EPA region or larger basins. Additionally, data from the surveys are available on our website, and we're looking to provide additional data from surveys very soon. You can also access our field and lab manuals online to learn more about how we implement those processes, as well as well as see many of the communication materials we've developed. Working to develop other tools we all hope to make available over the next year or so. So any feedback you have on the materials that you find or things that you think would be useful would be great for our team, so please feel free to let me know. Thank you so much for your time. If you have any comments or questions related to NARS, would like assistance accessing NARS data or tools, or if I can assist in any other way, please don't hesitate to reach out to me. Again, I'm very excited to learn about the work you all are doing and to consider ways to leverage NARS in support of your efforts. Thanks.